Hello, distinguished members of the Google organization. My name is Logan Weitz, and I am a junior at Long Beach High School on Long Island. I am part of the science research program here at Long Beach, and I have been for the past 3 years. In this presentation, I would like to take you through my life in the past 26 months. Now, before we begin, I must say that all research was conducted at the Weill Cornell Medical Center in New York City, and all human cells were taken from legal cell lines. Now lastly, this is not a curing cancer project, instead a cancer prevention experiment. Now, let us begin. The human p53 gene is a tumor suppressant. It is located on the short arm of the 17th chromosome. The p53 gene is responsible for the regulation of the cell cycle. It is known as the guardian of the genome. People who inherit only one functional copy of the p53 gene will most likely develop tumors in early adulthood. More than 50% of tumors recorded each year contain a mutated copy of p53. If a human female inherits the p53 gene, then she will have an 87.36% chance of contracting cancer, and a human male has an 85.88% chance of contracting cancer in one of the regions where p53 has been linked. These include heart, brain, liver, pancreatic, breast, and esophageal cancer. Upon my research about p53, I began to ponder if one could stop the inheritance of cancer instead of trying to treat it after contraction. I thought of this because my family only has one functional copy of p53, and while my mother was being treated for breast cancer, I hypothesized that the removal of the p53 gene from a family's genome will in turn stop the inheritance of cancer. But this could not be established unless a new gene could be placed in p53's place. The next step was trying to find that gene that could replace the p53 within the cell without causing further risk or rejection by the cell. Over the span of 18 months, I tested hundreds of genes from 13 different species of mammals. And astonishingly, I found the compatible gene within the trout, the Oncorhynchus mycus. The astonishing find was amazing. The PVF21 trout gene is located on the 3rd chromosome of the trout and is also responsible for the regulation of the cell cycle. While testing the PVF21 with human cells, I found that the gene does not cause further harm to the cell and fulfills the functions of the removed p53. In order to get the PVF21 into the human cells after the removal of p53 via the James chemical buffer, I used oligonucleotides, which are small single-stranded DNA that can take the gene directly to the nucleus of the cell. I did not use viral vectors in order to reduce the possible damage to the cell. Once PVF21 was successfully placed within the human pancreatic and liver cells I tested, I began to grow 4 separate cell cultures. These cell cultures were divided into Group A, Group B, Group C, and Group D. Group A was pancreatic cells that previously contained an unmutated copy of p53. Group B was pancreatic cells that contained a mutated copy of p53. Group C was liver cells that previously contained unmutated copies of p53, while Group D was liver cells that previously contained mutated copies of p53. Post-proliferation of each culture, I recorded the results and observed that each culture grew in size without mutation, as well as the concentration of PVF21 increased within each culture. These astonishing results show great possibility for the gene therapy field that could now change the family's genome for the betterment of the family line, so that we can help stop the inheritance of cancer within each family. I thank you for your time, and I truly do hope that you have seen the importance of cancer prevention and the potential that this procedure has in helping the millions of families that do not deserve the lifetime of helplessness and despair that cancer brings with it. Thank you, and have a great day.